So thank you for joining us. Today we have Nadine with us to share her latest research from Dr. Lisa Henske's lab. I had the honor of actually hearing about some of this research last November when I visited the Henske lab. And I was extremely impressed with Nadine's, not only her excitement about the research, but also making a difference and an impact for those affected by rare kidney cancers and chromophobe kidney cancer. So I couldn't be more excited to have you here to share your work with us and the community. The one thing that I want to also mention for those listening is that this research is preclinical, so it's not directly applicable to patients today. But the findings from understanding the biology and some of the mechanisms described in this paper is really helpful in understanding how Chromophobe works and identifying potential therapeutic targets to bring new treatments for Chromophobe patients, hopefully in the future, which I'm sure we'll talk about at the end as well, too, talking about how that can translate into new treatments and research going forward. So welcome. And can you briefly introduce yourself and share what drew you to studying Chromophobe kidney cancer in Dr. Henske's lab?
Thank you so much, Katie, for this amazing introduction. I'm very happy to be here today and to be talking to you again. And I'm going to take a moment to introduce myself. My name is Nadine Mahmud. I'm a medical doctor. Throughout my medical school, I was very interested in oncology, and specifically, I had a particular interest in rare tumors. And that pushed me to dedicate 2 years after my graduation to study chromophobe kidney cancer at Dr. Hanske's lab in Boston at Brigham and Women's. During these 2 years, I got to learn a lot about the biology and science behind Chromophobe RCC and to work a little bit on trying and testing some of the drugs in Chromophobe RCC. I've just recently moved to my internal medicine residency training at St. Louis University, and I'm very excited to be talking to you today.
I appreciate the fact that you had an interest in rare kidney cancers and rare cancers in general, just because I feel like it's such a neglected area that we need more people like you that are passionate about it. And one of the first things that I really found fascinating about your research and really sparked my interest when we were talking about it is that your research kind of highlights a different pathway and cell death than we've typically been used to hearing about, especially out of Dr. Henske's lab over the past few years. I know when your paper actually initially came out, we do have patients in the chromophobe community that follow those papers very closely. And somebody had actually even mentioned, does this mean that ferroptosis didn't work out? So I'm really excited to kind of bring the full details and be able to have you share about kind of the full approach that you took with this research and how it doesn't mean that ferroptosis didn't work out. This is actually showing the power of being able to combine effects and explore multiple pathways because cancer is so complicated. There's not always just one off switch. There actually almost probably never is just a single on-off switch. And so your research really highlights that. And I'm excited to hear more about it and share it with the community here. So to kick us off, can you explain to us what apoptosis is, which your paper highlights, and the rationale behind researching this in chromophobe kidney cancer?
Yeah, so sure. Basically, as you mentioned, we are still very interested in understanding ferroptosis and chromophobe RCC and trying to target it. And this is still being actively done at the Henske lab as of now. But we've been trying to do all kinds of stuff to induce cell death in chromophobe RCC. One of them is the most well-known form of cell death, which is apoptosis. Apoptosis has been historically studied as the primary form of cell death in the human body. All the time, we have all our cells that are dying and some others are regenerating at the same time. So apoptosis is a regulated form of cell death that is commonly used when there's any unneeded or unwanted or damaged cells in our body. And I said it's regulated. And the reason why I said it's regulated because there's a family of protein called the BCL-2 family of proteins that takes control over deciding if the cells should die or not at every moment. And this is very interesting because this family of protein is constituted of two families, actually. One of them wants the cell to survive, and it's called anti-apoptotic protein. And then we have another branch which wants the cell So to die, and these are called pro-apoptotic proteins. So all the time, at any time, we have a balance between these two families to either let the cell survive or let it go into apoptotic cell death. So why we care, the question is, why do we care about this in oncology and specifically in chromophobe RCC? The reason why is that sometimes tumor cells can take this to their advantage and can dysregulate this balance, I would say, between these two families to help the cell survive and overcome all stress and any immune cells trying to induce death in the tumor cell. So basically that happens through upregulating the anti-apoptotic family of protein, which is constituted by many members, including BCL-XL, BCL-2, MCL-1, and others. So these are just names of the anti-apoptotic proteins. We wanted to see if in chromophobe RCC there is any such thing happening, if there's any upregulation of any of these members. And the way to do that is to use an assay. It's a screening assay called the BHT profiling. The BHT profiling, although the name is very fancy, but the idea is very simple. We try to inhibit each one of the anti-apoptotic proteins one at a time. And then we try to see if the cell will die if we remove that member or not. If it does die, we can read that out through increased cytochrome C. Cytochrome C is a hallmark of apoptosis. It's a sign for us that there is incoming apoptosis now. And the BHT profiling results showed that there was a specific dependency in chromophobe RCC on a protein called BCL-XL. And I'm saying dependency because every time we inhibited this specific proteins, we saw that the cells were dying. So this was a good fundamental base that we use in our future studies.
As I was reading this paper from a patient perspective, I saw all of the letters and the acronyms for everything, right? And was trying to understand what they all mean. And so I really like how you explained that they're kind of seem like different sides to deciding if the cell is going to live or die. And if you have more of one or the other, possibly kind of overexpressing, you can imagine that would kind of take over. And so when you kind of turn one of them off, in my mind, what I'm kind of picturing is like now you've got a team, you're playing a game, and you've got 3 against 2 kind of a situation. Does your research kind of touch on that at all? Is that part of what the exploration was here to see if there might be reasons why it could be blocking apoptosis through the different avenues that trigger apoptosis?
Yeah, exactly. So the reason why we wanted to understand apoptosis, we wanted to see how far or how close chromophobe RCC tumor cells was from undergoing apoptotic cell death. So this PHC profiling that we use in our 4 chromophobe RCC cell lines. So these 4 cell lines are derived from human chromophobe RCC tumors from patients, and we cultured them basically in the lab, and then we use them for all our experiments. And we did this PHC profiling in all of our cell lines, the 4 cell lines, and we saw consistently that the cells were far from death. So they were resistant from undergoing apoptosis, and then they were not as ready as we would expect or hope for them to undergo apoptosis. And when we treated the cells with, for example, a molecule called BAD or another called HRK, which are supposed to inhibit BCL-XL, we saw that the cytochrome C release, which is the landmark for apoptosis, was increasing. And we saw that the cells were undergoing more and more apoptosis, the more we inhibited them, inhibited the BCL-XL in them. However, when we inhibited other proteins like the BCL-2, for example, we did not see that. So all of that to say that BCL-XL was a specific dependency in chromophobe RCC. And then, of course, this is a screening assay, so we wanted to confirm and validate these results. So we used a data depository called the TCGA, which is short for the Cancer Genome Atlas. And this is basically a database that has all the sequencing data for so many tumors. And it helps us compare the gene expression levels between different tumors to understand the differences between different tumors and compare them to normal tissue. And we use the TCGA data to study the gene encoding for BCL-XL. So the gene encoding for BCL-XL is called BCL-2L1. I know there are so many names as we talk, but basically we found something very interesting is that BCL-2L1 is fourfold upregulated in chromophobe compared to normal tissue. And whenever we see an upregulation, we would think and assume that there's a reason why it's upregulated. The cell cares about it being high. The cell needs it to survive. The tumor cell needs it to survive. And one more interesting thing is that we found that BCL2L, that chromophobe RCC had the second highest expression of BCL2L1 compared to all tumors in the TCGA data. Just again, strengthening and solidifying the idea that BCL-XL is a specific dependency in chromophobe RCC. So what we did, the next step was to inhibit the protein and see what happens if it's so essential and important. What would happen if the cell does not have any BCL-XL? And so basically, we use a genetic knockdown. We knock down the gene, which makes sure that there is no protein synthesized in the cell, because if the gene that is encoding the protein is not there, the cell would be empty of BCL-XL. And we saw that there was increased cell death in cells that had BCL-2L1 knockdown. But interestingly, one more thing happened. We studied the protein levels of all the anti-apoptotic proteins to see if the cell is reacting in any way to fight this alteration that we did in the cell. And we found that there was an upregulation of another anti-apoptotic protein called MCL-1. And this is like a way of the cell to fight and resist the lack of BCL-XL. It's like we shut one door, it opens another. So our thought process was then we can shut the other door as well and see what happens. And we literally inhibited both BCL-XL and MCL-1 at the same time. And we saw that there was almost around 80 to 100% cell death in the cells that didn't have any BCL-XL or MCL-1. It's like the tumor cell was not able to fight anymore. So this was very interesting for us. And we also validated this finding from also the DepMap, which is another data repository that also showed us that this has been seen in other kinds of tumors. Yeah, so I think this was very interesting for us to see.
That's really exciting. So, if I'm understanding this correctly, and please correct me if I'm wrong, but so we already know from previous research about chromophobe kidney cancer that it's highly sensitive to ferroptosis. So that it's kind of sitting on the edge, just waiting to be pushed into cell death from ferroptosis. And so that's one area where we knew that it's kind of close to that edge. Just figuring out how you push it over, I think, is the areas that there's a lot of active engagement in exploring. But then with apoptosis, that's kind of on the other side, a different type of cell death. And that was the complete opposite that you observed from these cells just in their natural state of they kind of showed no signs to being close to that edge at all. But the research that you have done basically finds if we go back to the analogy of the like 3 versus 3, like you have those proteins that are kind of competing against each other, and then you knock one of them down, which was the BCXL. Then you have 3 versus 2, and it pushes it closer to the edge, but then it starts. Maybe player number 2 starts outperforming and trying to pick up the slack
Exactly.
for the missing player. And so you then knocked that one out as well too. There's still a little bit of activity, but it's kind of knocked most of it down.
This is exactly how we think about it. And we think that these two players that we knocked from the team are the two most important. And again, this is just an example, but again, to remember there are other team members that we're not right now discussing, but they are there. So
Yeah.
yeah, exactly what you said. These are the main players that we care, that the tumor cell cares about, and now they're gone.
You then looked into if there are drugs that may be targeting these proteins. So is there anything that can influence this, maybe to take it to the next step outside of the research that you're currently doing in the lab? Can you share what findings you explored in the paper through that?
Yeah, so the interesting thing is now we have this piece of information that we know these proteins play a role, but then there are more directions we need to follow before any of that can reach human trials. As in any other drug, FDA approval, there are certain steps that needs to be done. Now we're done with the basic lab side, and there will be the animal in vivo work where we test these medications in animals and see if they're safe in terms of adverse events, toxicities, if it works in a body system rather than just a cell. And if all of that work, then these can be advanced into clinical trials, which would be very interesting if that happens. But again, there are not all the drugs that we use in our studies are actually in clinical trials or are candidates to be there. So we basically tested many ways of getting rid of BCL-XL. One of the ways was what we call a PROTAC or a proteolysis targeting chimera. And this is not a direct inhibitor. So a direct inhibitor would literally sit on the protein and inhibit its action. It's like blocking the protein from doing its job. However, the PROTAC or proteolysis targeting chimera, it's a new technology that's starting to be commonly used in cancer and other treatments, where the drug that we give tags the protein of interest, in our case, BCL-XL, for elimination. And it's basically literally just putting an X on the protein, telling the cell that we need to get rid, you can trash down this protein, we don't need it. So it's synthesized, but it's not active. And this presents an advantage to other inhibitors, because you're probably asking, why do we need the PROTAC if they are inhibitors? The inhibitors have been associated with so many platelet toxicities because platelets have a lot of BCL-XL. So any inhibitor that we would use has so many side effects in terms of platelets, low platelets and stuff. And so we moved to the PROTAC that are safer in that aspect. So the PROTAC there are no FDA approved PROTAC so far for anything, but there are DT2216 that we use in our studies. It's a BCL-XL targeting PROTAC. It was in phase 1 clinical trial that has been just completed, and we're still waiting for the results of this trial that has been tested in other tumors. We also tested another PROTAC called the XZ739. Again, so many names, but basically, this is just another PROTAC. It's not in clinical trials, but hopefully, we're going to be seeing more studies using that PROTAC before it's escalated into trials. But the S63845, which is an inhibitor of MCL-1, has been shown to be very toxic in mice and was never actually tested in humans. So all of that to say that there are still a lot of work to be done, but hopefully we are very positive and hopeful that we're gonna at least have the BCL-XL PROTAC tested at some point.
Yeah, I think that's what I find so important about the basic science research is because it really helps understand the biology, what's happening kind of behind the scenes. And then patients, when it comes to patient care, it's so far down the line of having treatments that have been tested in all of these different avenues. But if you understand the basic science, what the key components are, then you can keep your eye out for these new trials and treatments and areas that have been explored by other groups to see if there are treatments coming on board that you might eventually be able to try for clinical practice. Again, it takes a long time to get through that process, but I think it's exciting for me as far as knowing that the basic science is the one that helps kind of point the direction instead of just like picking a treatment with chromophobe kidney cancer and say, we hope this works. It makes it so that I'm hopeful that in the future there'll be more targeted clinical trials that really represent the biology of chromophobe kidney cancer because it's something that we are learning a lot more about. Yeah. And I think that's a good segue into kind of the second part of your paper, which really highlights the synergies with ferroptosis. And that's where I also found this research very exciting because I've been following some of the other papers that have come out of Lisa Henske's lab and have been following ferroptosis for quite a while and know that some of the medications used to trigger ferroptosis can also be pretty toxic because you need such a high dose that it introduces a lot of toxicities. And so, from reading your paper, I really enjoyed the fact that you tried to synergize that and see if you might be able to explore targeting both ferroptosis and apoptosis at the same time. So, I was wondering if you could share a little bit about what that research was, what made you want to explore it, and the findings from it.
Yeah, so I feel like it's a surprising section when you're been talking about apoptosis all the time, and then you're like, okay, we're now talking about ferroptosis. The rationale behind it is when we started working on inducing apoptosis in chromophobe RCC, we were very satisfied to see that there are so much cell death by just inducing apoptosis. However, talking to other specialists in apoptosis, we realized that in other tumors, inducing apoptosis has been used as a complementary thing on top of the standard of care treatment. For example, the advancement in inducing apoptosis have been usually coupled rather than just being the sole treatment that patients take. It's usually on top of the standard of care in cases of refractory, resistant tumors, or like patients with really advanced and metastatic disease. So we thought, like, what can we use these apoptosis inducing agents with? And the question was very hard because, as you may know, there are nothing we can use. So we tested a range of drugs that, like rapamycin and other medication that we know patients with chromophobe RCC have been using, and we didn't see that there was any crosstalk between them. So we were not able to see that these attacks and everything were adding any benefit to rapamycin, for example. But then we were like ferroptosis inducing drugs like IKE and RSL3 have been showing very promising results in vitro and like in our lab. And so we thought, why not try apoptosis and ferroptosis? So we combined these two medications, the apoptosis and the ferroptosis inducers, and we were very happy and surprised to see that there was a certain crosstalk and there was way more effect with just some concentration, very low concentrations of the IKE or RSL3, which are the ferroptosis agents, inducing agents. So we were very excited about this. We wanted to understand more about it. So we started literally just inhibiting BCL-XL and then studying the protein levels, like the levels of proteins that are related to ferroptosis, like SLC7 and 11. So these proteins, they are only involved in ferroptosis. So we know if there's any dysregulation in these proteins, we know that this is like a sign that ferroptosis is involved in what's happening. So we saw that whenever we induce apoptosis, the genes involved in ferroptosis were going up. So this provided like an extra sign that there was something going on between these two pathways. And then we literally just studied the levels of glutathione, which is also involved in ferroptosis. That's the whole idea behind ferroptosis, actually. And we saw that the reduced glutathione, which is the used antioxidant, was so much lower when we treat with the apoptosis inducer. So basically, we saw that the cells was using all the antioxidant there when we induce apoptosis. So there are definitely so much still to be done in that area to understand how this is happening. But what our study suggests is that there is a crosstalk between these two pathways, and there's a lot more to explore there. But I find it very promising that these two drugs work hand in hand.
I can imagine that would potentially mean that you could have lower doses of these for a combined treatment that is less toxic and more tolerable to patients and actually hopefully receive a response. Is that kind of the hope there in exploring the synergies?
Yes, exactly. As you mentioned, we always care a lot about doses. So sometimes things work, but the fact that that higher doses are needed for them to work just makes it an issue because these are dose limiting toxicities and issues that come up with higher doses. So I think the fact that the dose of IgE and RSA3 that were required to actually induce so much more death were very minimal compared to what we usually use with like if you're using just IKE and RSLC by themselves. So yeah, totally, yes.
Very exciting. So, as we wrap up here, my final question to you is if you had a message you could share with patients that are affected by chromophobe kidney cancer, what would that message be, or what are you most hopeful for the future of patients who've been affected by chromophobe kidney cancer?
Yes, in terms of my message to patients with Chromophobe RCC, I want them to know that they are not alone. And I know the disease is rare, and at many times patients maybe feel isolated because maybe their doctors have not seen other patients with chromophobe before, or have never treated patients with chromophobe before. But what I want them to know is that there are dedicated and growing communities of clinicians and scientists and biologists and physicians working very hard behind the scenes to change this fact. I am very hopeful about the future of chromophobe RCC, and I'm very hopeful that in the future hopefully we're gonna be looking at treatment options that are specifically tailored to the biology of chromophobe RCC as we understand it more and more day by day. Apart from the research, I want patients with chromophobe RCC and other rare kidney tumors and other rare cancers to know that they are heard, and their voices matter. And we are, as scientists or physicians or anyone working on chromophobe RCC, we are very inspired by them and by their strength. And this is what pushes us every day to keep working and hoping that one day chromophobe RCC is just going to be just like any other tumor, just as treated, just as understood, just as studied. So hopefully that's the future of chromophobe RCC.
Yeah, thank you so much. That's such an important message for patients. And thank you so much for your research and your part in this, because you are moving that forward and you are providing that hope. So thank you for being a part of that as well too.
Thank you, Katie. We are very grateful for you too.
Thank you for your time here today. We will share a link to the paper in the description below.
All right. Thank you so much.